Leading our coverage this week is a significant developments in global public health following a cluster of severe respiratory illnesses and deaths linked to a specific viral discovery. Officials from the World Health Organization and the Centers for Disease Control and Prevention have confirmed that a rare and highly lethal strain of hantavirus is behind a multi-country outbreak first detected in May 2026. This report follows the release of genomic data that points to a specific mutation in the virus's architecture, a development that scientists are calling a genome mystery due to the speed at which the pathogen affects human lung tissue. The epicenter of the current investigation is the cruise ship MV Hondius, which departed Ushuaia, Argentina on April 1, 2026. The vessel was carrying 147 passengers and crew across the South Atlantic when a series of medical emergencies forced international intervention. As of early May, health officials have identified at least 8 cases, including 3 confirmed deaths, with a case-fatality ratio currently estimated at 38%. Plus 4 hantaviruses are a family of viruses spread mainly by rodents, and while they are found worldwide, the specific strain involved in this outbreak is known as the Andes virus. This is a critical distinction for public health experts because the Andes virus is the only known hantavirus capable of human-to-human transmission, a factor that complicates containment efforts in enclosed environments like ships or airplanes. The illness, known clinically as hantavirus pulmonary syndrome or HPS, begins with non-specific symptoms such as fever, muscle aches, and gastrointestinal distress. However, the progression of the disease is notoriously rapid. Within days of the initial prodromal phase, patients often experience a sudden onset of respiratory distress as their lungs fill with fluid, a condition that the latest reports suggest may be accelerated by specific genetic variations in this year's circulating strain. Plus 2, the geographic footprint of this outbreak is unusually broad due to the itinerary of the vessel and the subsequent travel of its passengers. After leaving Argentina, the ship visited remote locations including Antarctica, South Georgia, and St. Helena. One passenger, who later died, carried the virus onto a commercial flight to Johannesburg, South Africa, prompting a massive contact tracing effort across multiple continents. Public health framing for this story emphasizes that while the risk to the general public remains low, the clinical severity of this particular hantavirus mutation has put diagnostic labs on high alert. The discovery of these viral changes comes at a time when researchers are trying to understand why some hantavirus cases lead to mild illness while others result in total pulmonary collapse within 48 hours of the first cough. This report will look at the specific mechanisms of this mutation and what the global surveillance network is doing to prevent a wider spread of what is traditionally a localized zoonotic threat. At the heart of the ongoing scientific investigation are several high-security laboratories, including the National Institute for Communicable Diseases in South Africa and university hospitals in Geneva, Switzerland. These facilities have been working around the clock to sequence the viral genome from the victims of the cruise ship cluster. The core development in this case centers on the L segment of the Andes virus genome. Preliminary analysis has identified 2 specific single nucleotide polymorphisms, or SNPs, that distinguish this 2026 strain from historical samples collected in Argentina decades ago. Specifically, mutations at positions C2139T and G576A have been labeled as true viral mutations rather than artifacts of the testing process. While these are currently categorized as synonymous mutations, their presence in such a high-fatality cluster has led researchers to investigate if they influence the virus's ability to replicate or evade the human immune system. Plus one further complicating the mystery is research released by the University of Texas at Austin just prior to this outbreak. Using advanced cryo-electron microscopy, scientists mapped the highest resolution molecular structure ever recorded of the virus's surface protein complex, known as the GNGC tetramer. This mushroom-shaped structure is the key mechanism the virus uses to latch onto and infect human cells. The study revealed that hantaviruses utilize beta-3 integrins, receptors typically involved in regulating vascular permeability, to gain entry into the lining of the lungs. The instant shutdown described in recent headlines refers to a catastrophic cytokine storm and vascular leak syndrome. In this process, the virus does not kill lung cells directly. Instead, it triggers a massive overreaction from the host's immune system. This causes the pulmonary capillaries to become porous. allowing fluid to flood the air sacs and effectively drowning the patient from the inside. Plus one laboratory teams are now contrasting these structural findings with the 2026 cruise ship data to determine if the detected mutations make the virus more efficient at triggering this fluid leakage. The current evidence suggests that the outbreak likely began with a single zoonotic spillover, a passenger exposed to rodent droppings or dust while hiking in South America, which then transitioned into limited human-to-human spread on the vessel. This core development is significant because it marks one of the few times that modern genomic sequencing has been able to track an active hantavirus transmission chain in real time across international borders. While the number of cases remains small, the data density provided by these labs is helping to refine the blueprint for potential vaccines and antibody therapies, which are currently non-existent for this family of pathogens. As the international community monitors the remaining passengers from the MV Hondius, the focus has shifted to prevention, surveillance, and the sobering reality of medical limitations. Public health guidance remains consistent. There is no cure, vaccine, or specific antiviral treatment for hantavirus pulmonary syndrome. The primary strategy for survival is early supportive care. For the most severe cases, clinicians are turning to extracorporeal membrane oxygenation, or ECMO, a process where a machine pumps and oxygenates a patient's blood outside the body, allowing the heart and lungs to rest. Data indicates that when ECMO is initiated early, survival rates for HPS can climb to 80%, though the availability of such specialized equipment is limited in the remote regions where hantavirus often emerges. +2 for the general public and travelers, prevention is centered on rodent control and environmental hygiene. Because the virus is typically shed in the urine, saliva, and feces of infected long-tailed pygmy rice rats and other rodent reservoirs, officials advise against sweeping or vacuuming areas where rodents may have been present. Such actions can aerosolize the virus, creating a fine mist that is easily inhaled. Instead, the CDC recommends soaking contaminated areas with a mixture of bleach and water before cleaning. In the context of the current outbreak, the World Health Organization is emphasizing that while the Andes virus can spread between people, it usually requires close, prolonged contact with a symptomatic individual. This includes sharing utensils, physical intimacy, or spending extended periods in poorly ventilated, enclosed spaces. +1 surveillance networks are now operating with an honest admission of uncertainty. It is not yet clear if the mutations discovered this month represent a permanent shift in the virus's biology, or if they are simply a snapshot of a single tragic event. Public health agencies are continuing to track 147 individuals across 23 different countries to ensure no further secondary transmission occurs. The limits of our knowledge are clear. We are still learning how these viruses jump from animals to humans and why the human immune system reacts so violently to the infection. As researchers at UT Austin and other institutions move closer to a stabilized vaccine candidate, the 2026 outbreak serves as a reminder of the speed at which zoonotic diseases can move in a globalized world. For now, the situation is being managed through strict isolation and rapid diagnostic testing. We will continue to follow the scientific consensus as more genomic data is released. Subscribe for further updates on global health reporting. The current investigation into the reported hantavirus mutation has centered on high-containment biosafety Level 4 laboratories, where researchers are analyzing the virus's complex genetic architecture. To understand the gravity of the recent discovery, it is necessary to look at the three-segmented negative-sense RNA genome that defines this family of viruses. The genome is composed of the large or L segment, which encodes the viral polymerase, the medium or M segment, which encodes the surface glycoproteins, and the small or S segment, which encodes the nucleocapsid protein. Scientists monitoring the recent cluster of cases are focusing specifically on the M segment. This portion of the genetic code is responsible for the formation of the GN and GC glycoproteins, which assemble into a spike-like structure on the viral envelope. It is this specific molecular machinery that allows the virus to recognize and bind to human cells, particularly the endothelial cells lining the pulmonary capillaries. Recent genomic sequencing has identified subtle variations in these spikes that may enhance the virus's binding affinity, potentially explaining the rapid clinical deterioration observed in recent patients. Public health agencies, including the Centers for Disease Control and Prevention and the World Health Organization, are currently cross-referencing these new sequences against historical databases of the Andes and Sin Nombre strains. The primary concern among virologists is the mechanism by which the virus induces what has been described as a sudden shutdown of the respiratory system. In a clinical setting, this is recognized as Hantavirus pulmonary syndrome, or HPS. Unlike many other respiratory viruses that cause direct tissue destruction, hantaviruses are generally non-cytopathic, meaning they do not kill the cells they infect. Instead, the June mystery currently under investigation involves how the virus triggers an overwhelming immune response. When the virus enters the lungs, it targets the beta-3 integrin receptors on the surface of endothelial cells. This interaction initiates a massive release of pro-inflammatory cytokines, such as interleukin-6 and tumor necrosis factor alpha. This cytokine storm causes the junctions between capillary cells to fail, leading to massive vascular leakage. The lungs essentially become saturated with the patient's own plasma, resulting in hypoxia and myocardial depression within a matter of hours. The reports of an instant shutdown are being contextualized by medical officials as an exceptionally short incubation to emergency window. While the initial flu-like prodrome can last several days, the transition to the cardiopulmonary phase is notoriously abrupt. Laboratory findings from recent cases have shown an unusually high viral load in the early stages of infection, leading to theories that the mutation may allow for more efficient viral replication within the host. Testing protocols at specialized clinics now prioritize the identification of immunoblastic fragments in peripheral blood smears, a hallmark of hantavirus infection that can appear even before severe respiratory distress begins. Surveillance teams are also tracking the distribution of the rodent reservoirs, such as the deer mouse in North America and the long-tailed pygmy rice rat in South America, to see if environmental factors or mutations within the rodent populations are contributing to the increased virulence. The scientific community remains cautious about labeling these genetic shifts as a definitive change in the virus's fundamental nat-ur. Historically, hantaviruses have shown significant genetic diversity due to their reliance on specific rodent hosts, and what appears to be a new mutation may simply be a previously undocumented variant emerging in a high-density human setting. National health institutes are currently contrasting these laboratory findings with real-world clinical data to determine if the mortality rate is truly deviating from the historical average of 35 to 40%. As the genomic data continues to be uploaded to international repositories like GZAID, the focus remains on the structural biology of the virus. Understanding the exact configuration of the GNGC glycoprotein complex is considered the holy grail for developing neutralizing antibodies. Until these lab results are peer-reviewed and replicated, officials are maintaining a stance of heightened surveillance rather than alarm, emphasizing that while the virus's mechanism is devastating, its transmission remains largely tied to specific environmental exposures. In the wake of recent findings regarding the potential hantavirus mutation, the global health community is pivoting toward a dual strategy of aggressive environmental prevention and enhanced clinical surveillance. For the individual, prevention remains the most effective tool, as medical interventions after the onset of the pulmonary phase are historically limited in scope and success. Public health officials emphasize that hantavirus is primarily a zoonotic disease, meaning it originates in animals, specifically rodents like the deer mouse, the cotton rat, and the long-tailed pygmy rice rat. The virus is shed through the saliva, urine, and droppings of these host animals. Transmission to humans occurs most frequently through aerosolization, a process where dried viral particles are disturbed and kicked up into the air to be inhaled. This is why official guidance is explicit. Never use a broom or a vacuum cleaner in an area where rodent activity is suspected. Such actions propel the virus into the breathing zone of the individual. Instead, the protocol involves saturating the area with a disinfectant, ideally a 10% bleach solution, for at least 5 minutes before wiping it down with damp paper towels. This method ensures that viral particles remain trapped in the liquid and are chemically neutralized before they can be disturbed. On a broader scale, surveillance efforts are being intensified through a One Health approach, which recognizes the interconnectedness of human, animal, and environmental health. In regions where the recent cluster of cases originated, field biologists and epidemiologists are trapping and testing local rodent populations to determine if the viral load or the prevalence of the mutation is increasing in the wild. This early warning system allows public health agencies to issue localized alerts before human cases emerge. However, there are significant limits to our current knowledge. The Genome mystery highlighted by recent reports underscores a critical gap in virology. We do not yet fully understand why specific mutations suddenly increase the virus's ability to shut down human lung function so rapidly. While the genomic data provides a blueprint, it does not immediately provide a cure. There is currently no FDA-approved vaccine or specific antiviral treatment for hantavirus pulmonary syndrome. Clinical management is limited to supportive care, often requiring the use of extracorporeal membrane oxygenation or ECMO. This intensive procedure involves bypass machinery that oxygenates the blood outside the body, but its availability is often restricted to tertiary care hospitals in developed urban centers, creating a significant ethical and logistical challenge for treating patients in the rural areas where hantavirus is most endemic. The reporting on these mutations also brings to light the necessity of managing public perception with scientific accuracy. While the term instantly shuts down lungs conveys the clinical speed of the disease, it is technically an immune-mediated collapse rather than a direct viral attack on the tissue. This nuance is vital for researchers developing immunotherapies designed to dampen the body's overreaction rather than just attacking the virus itself. As the investigation into the 2026 strains continues, officials are maintaining a stance of cautious transparency. The uncertainty surrounding the mystery mutation is a standard part of the scientific process, where hypotheses must be tested in controlled laboratory environments before definitive conclusions are drawn. For travelers and those living in rural or semi-rural areas, the advice remains grounded in physical barriers. Seal entry points in homes to keep rodents out, keep food in thick plastic or metal containers, and use professional-grade respirators when cleaning long-abandoned outbuildings. By combining these individual precautions with international laboratory cooperation, the goal is to contain this highly lethal pathogen before it can establish a broader foothold in the human population. The situation remains under constant review by global health authorities. For more in-depth reporting on emerging public health threats and genomic science, please consider subscribing to our channel.